Hello everyone. Was not expecting such a big room, but thank you for having me here. And my name is Priyanka. I work for the Tree of Life consortium at the Sanger Institute. We're part of a global initiative to sequence all complex life. It's called the Earth Biogenome Project. We're directly involved with the Vertebrate Genomes Project, the European Reference Genome Atlas, Aquatic Symbiosis Genomics Project, but our main project is the Darwin Tree of Life, which is going to sequence over 70,000 different plants, animals, fungi, and protists over the next 10 years in Britain and Ireland. And we cannot do any of this work without all of our partners, which are all working together to help understand the biodiversity of life, create new tools for genomics research, and also identify new biomolecules from all these new organisms for medicine and biotechnology. In all the work that we do, we try to follow fair data practices, making our raw data, genomes, and downstream analysis results available rapidly without any embargo on easily findable public databases. We also have portals where anyone who's interested in our data can track them. So we have a Tree of Life quality check portal where you can track the progress of our assemblies and data. You can also look at our QC values for sequencing and cobint information. And we have a second portal called Genomes on a Tree, GOAT, which is a metadata aggregator, but it also keeps track of how far we've made it to that 70,000 mark and beyond. But at this scale, when we're working with this scale of data, we want to create like a completely automated engine, which will be able to track samples all the way from acquisition, which could be in any part of Britain and Ireland, through barcoding, extraction, sequencing, all the way through quality assurance. We have an internal STS benchling system for that, but beyond that point, starting from quality assurance, that's where the informatics pipelines come in, and they go through assembly, scaffolding, curation, submitting to ENA, where our partners at Ensemble do the annotation for us. Coming back to Sanger, where we do some more downstream analysis to create automated genome publications, and then a further analysis for comparative genomics. And we started this process earlier this year, and we decided to do it with Nextflow DSL2 and using NF-core toolings and templates. And that has been really very helpful for us because most of us were completely new to Nextflow. So having those tools made the transition very easy. Having those modules made it very easy for us to quickly build up our pipelines. All the pipelines that we have in development and that are in production are available on our GitHub website at Sangatol. So you can access it, you can comment on it, you can contribute to it, you can ask us to add features to it. We're happy to take all those things into consideration. We've started doing some work on scaffolding and curation, but most of our focus has been so far on getting these automated genomic publications. So that's what I'll talk about mostly today. So these are brief publications. They are actually citable, peer-reviewed publications that summarize all of our data results and are published on Wellcome Open Research, which is part of F1000. Underlying this publication, there are a series of these NF-core-based pipelines, but we're not currently part of NF-core. Similar to Bactopia, we're maintaining our own versions. So we start with a series of asynchronous pipelines which download data. So we have INSDC download, which brings data from NCBI. And then we have pipelines for repeat and gene annotation downloads. And these pipelines, they work based on the database timeline. So it could be that I get the genome sequence today and then repeat annotation 2 weeks later, and then gene annotation another few weeks later. So we have a database that keeps track of all this, and once we have the requisite input data, we move on to the next stage where we do read mapping, which is aligning our raw reads to the genome, and we work with lots of different sequencing technologies. PacBio, Illumina, 10x, I think we're the only ones working with that. We do Hi-C. We're also trying to add BioNano and RNA-seq functionality here. And for our internal work, we have a sub-workflow that automatically takes ID and project value and creates the sample sheet. For external data, anyone can provide a sample sheet and do the work. We'll soon be adding sequence composition functionality to this as well, which is things like telomeric and centromeric repeat, GC content, and so forth. Once we have all the alignments done, filtering, we move to the next stage where we have a pipeline called Blob Toolkit. It creates blob plots based on completeness, QC values, a coverage, cobient, and finally, the genome node pipeline integrates all these results along and creates contact matrices using Hi-C data. And we add more tracks to it to make increased value of the product, such as mappability. As we are sequencing more and more individuals for all these different species, we will also be adding species variant calling to it, and all this gets integrated using a predefined XML to create these automated genome nodes. These pipelines are going to be the backend of our portal, where anyone around the globe can submit their verified. So generally, even if it is private, as long as it's in a verified database like ENA, you can submit your FASTA file and your location and your raw read location, and the genome node will be created for you. We do extensive testing on all of our pipelines to ensure that they're meeting the standards that NF Core community sets, as well as that we have for ourselves to. So we do unit testing first on both the cloud and our local server. We also do a proof of concept testing with a small fungal genome. And we do further tests with some medium-sized genomes and a large genome. Very soon, we are going to be doing edge case testing with the European mistletoe, which is 90 GB. It's one of our largest genomes, so it has broken a lot of tools when we initially were doing the assembly, and so we know it's going to break most of our pipeline. So it's a great edge case to work with. So yes, this has also allowed us, as we work with these diverse stacks, most of the software are designed for humans or specific organisms. It allows us to contribute back to the software, improve them, make them more open source, or bring it more to the forefront so we can pull it into the pipelines that we are designing. We use a series of databases and WestKit, which is a workflow execution service with native Nextflow and snake capability to deploy all of our pipelines. We do the production work, at least for internal samples on our local server, but we test all our pipelines in the cloud as well, so they are cloud friendly. And any work that we do with GPU processes and GPU-based pipelines, we use Google Cloud for that. Finally, I'd like to thank a lot of people, starting with NF Core for providing the tooling to us. Since I'm so new to Nextflow and NF Core, it has been really helpful. And everyone who's on Nextflow and NF Core Slack answering the questions. Recently, this year, we participated in Google Summer of Code, where we worked with Alexander, who helped us start the conversion of Blob Toolkit pipeline. And the lead developer, Rich and Sujay, they were very helpful in that. We have several informatics teams in Tree of Life that work on different aspects of these genomes. So we have the assembly teams that does the assembly curation. They also maintain the Tree of Life QC portal. We have platform teams which help us build the APIs and web apps. Our great infrastructure team, we do a little bit of everything. So pipelines, portals, data management, and finally, Wellcome and UKRI for giving us the money for this fun project. Thank you.
Great, thanks a lot. So I've got a couple of questions here. One of them is from Alex, is asking, could you envision using public sub-workflows from NF Core to contribute to these, now that there is support in them almost, and the available aid in having modules and sub-workflows from private and public repositories in your workflows?
Okay, I'm going to answer the first question
Yeah.
first. Okay, so for the sub-workflows, yes, I worked with the sub-workflows team this hackathon, and that was the main reason. I wanted to understand how they are named, how they will be structured, and how we can utilize it. For our pipelines, the green stuff is the ones where we are mostly or entirely using NF Core modules, but I'm hoping we can start using the NF Core sub-workflows, contributing back some of our NF Core base sub-workflows back as well.
Okay, cool. And then do you use a controlled vocabulary and schema for checking
Sorry?
do you use a controlled vocabulary and schema for checking of the annotations?
I didn't get the last part.
For checking the annotations, do you use a controlled vocabulary?
For annotations, we rely on Ensemble to do that. So they have their Ensemble Rapid Release, which is what they use for annotation. We download that. We also do some more detailed annotation on family representatives. So we have certain species which represents family in these different taxons, and for those, we do more detailed analysis using Breaker and some other tools.
And then how different are the pipelines across different species? Do you have to modify them a lot? Are you creating many different pipelines, or?
No, that's our main rule. We want a single pipeline for all our species. To make sure we use resource optimization based on genome length and certain other input values, we adjust the amount of resources that are assigned. That way we're not failing, repeating multiple times, but we want to stick with the same pipeline for everything.
And I guess the process then for the original sequencing, is that all standardized as well across different species, or do you have to use quite different techniques?
It depends from species to species. That's why we like to have multiple data type support for all of our pipelines. So by standard, we always do hi-fi sequencing, PacBio hi-fi and Hi-C sequencing. That's our base standard. But depending on this, species, we might even do nanopore in Illumina, BioNano. So that's why any pipeline we do, we try to support all possible data types and have certain parameters that can turn off the parts that are not present.
And then one question here is from Bruno. Are you aware of the reasons for using WestKit for the workflow submission as opposed to Nextflow's native execution engine?
So we do use NF Tower CLI, but WestKit allows us to chain these different Nextflow pipelines as well as deploy them as and when the data becomes available. So we are very dependent on when the data is coming from the sequencing machines as well as these different databases to launch our different pipelines.
Mm-hmm. And then there's a question from Conrad there, which is around, do you rerun the pipelines as data changes, or do you have in situations where you're updating the runs based on, I guess, new tools?
Yes and no. We do versioning, and we're very strict with what we count as versioning for our pipelines. If, as a team in the infrastructure team, we decide this is a serious upgrade, yes, we do run the pipelines, but we keep track of everything, even our old runs. If it is a minor update, then no. And the genome node can also be updated. So if we have a major upgrade, we do push it to the genome node. And within genome node, you can see different versions.
Fantastic.
Okay. Thank you so much.